everyone. Thank you for coming. My name is Connie Rich, and I am a scientific product manager and the operations coordinator for VectorBuilder. And I'm here today to introduce you to our latest contribution to the scientific community, VectorBee. This is a DNA visualization, editing, and analysis software that is intuitive and versatile and 100% free. Now, when we're talking about, let's see, there we go. When we're talking about DNA software, there is a wide range of experiments and applications that this is really important for, but it's a very crucial tool in gene delivery. Some of us may remember times when we would take large amounts of sequence data and just paste onto a Word document, spend a lot of time trying to plan cloning experiments before headed to the bench to try things out, to try to make the vectors that we needed. Now, luckily now we do have DNA software that allows for us to interact more with that sequence. So we can add things like annotations and restriction sites, but at the end of that experience, we still only have a plan. We still have to go to the bench in order to make those vectors a reality. Now, luckily, VectorBee not only allows for incredibly user-friendly and intuitive interaction with that sequence to make our perfect vector, but additionally, after making this beautiful vector, seamless integration with VectorBuilder allows for you to have this perfect vector in your hands in the form of a plasmid, a virus, mRNA, or an entire library with no bench work necessary, freeing you up to actually answer the questions for your research that only you can answer. Now, VectorBee is an offshoot of VectorBuilder that integrates a lot of the key components that people love about using VectorBuilder in the Vector Design Studio. We allow you to utilize vectors that you have previously made, or we've selected popular vectors, as we'll see when we go through the software shortly. You can also utilize the extensive databases that VectorBuilder has created of a wide range of components, and arguably most importantly, you can utilize VectorBuilder's not only expertise, but also ideal cloning and efficiency in order to actually create this vector and provide you with downstream services like virus packaging, as well as stable cell line generation and library construction. So I wanted to hit these highlights about how VectorBuilder pairs with VectorBee, but there's no better way than to actually show everyone how we can get into VectorBee and utilize all that it has to offer. So when we go into VectorBee after you've downloaded the software, from the homepage, you have a wide variety of ways to open various different sequence and vector files. So if you've created something in the past, you can select this open file, and we accept a number of different formats like .vb, .fasta, and .dna. You can also explore popular vectors that VectorBuilder has identified, things like control vectors and popular ORFs in a variety of different backbones. You can also paste an entirely new sequence, or you can import vectors from VectorBuilder, as we mentioned. So you can search by vector ID, or if you've created a vector in the Vector Design Studio, it automatically populates here so that you can just select which vectors you want to work with for that particular project and import them straight in. Now, once you have these vectors in VectorBee, they appear as various different tabs so that you can easily switch between these different projects and open exactly what you need, have everything ready to go. Now, when we have our vectors open, we can easily switch between different formats and visualization preferences. You can go from this sort of horizontal view into a vertical view, where in this case, you have the vector map that's along here on the top, and the sequence is just below. Now you can scroll through and find the relevant region of the sequence you want to work with, but a more efficient way is to just click on the various different components that you're interested in, and it will jump to that region of the sequence. Now, in this vertical view, one of the ideal things that you have that's specific to vertical view are these maps. If we click on Show Coding Map, this is going to show us excuse me, if we just stretch this down here, this is going to show us the +1, +2, and +3 frames for the forward strand, and the +1, +2, and +3 frames for the reverse strand. And if we zoom in here, what we can see are these little red and green boxes. So these are start codons, and for the red, these are stop codons that are present on each of these strands. Now we can view this either sort of globally or zoomed in. And similarly, we can see GC content maps to visualize GC content across the entire vector. Again, we can also zoom in on this as well. Finally, we can show CPG and GPC maps that show this dinucleotide frequency across our entire vector. And this can show us potentially interesting regions, give us insights into chromatin availability, things like that. Now, this is very useful if you're looking at these kind of global trends and kind of planning for those types of cloning experiences and potential issues. But I personally prefer this horizontal view right here. So in this horizontal view, you still have a lot of the same interactions. So we can still get the sequence, we can still click on individual features and jump to that region of the sequence, and we can still do editing capabilities for our individual regions of sequence. So this really fits the bill for most of what we're wanting to do. But we also have the ability to change the visualization for intensity of lines, for size, for spacing. Let's say I want to make my text a little larger, or if I want to make it a little smaller, you can apply this to all of your different projects to just fit your personal preferences. And now, additionally, for these types of visualizations, they're very pleasant to be able to just have what you want to look at, but the features themselves are something that are really important. Now, for these various different features that you see here, these are vectors that I imported from VectorBuilder, but whether you're importing or whether you're pasting a sequence from scratch, all of these features are going to be automatically recognized. But you still have a lot of customization available. You can create features, you can edit or copy, or you can go to this features list to see all of the features that are present in your sequence. You can view more details, or you can view things just in a standard list. You can perform actions like set default orientation in these bulk actions. You can change visibility, and you can edit individual features. So here I'll edit this EGFP. I could change the name, the orientation. I can change the type of sequence I'm looking at, as well as a number of different options. Here, I'm just going to add a note that I want to change this to SOX2 later on. So we can change all of these individual parts of this feature and kind of how it's visualized. But one thing that's not present in this sort of edit feature capability that I do want to highlight is that you can change parts of the feature. So how it's visualized on the map. So let's say I want to, in this EGFP, really highlight for my own reference where this tag, let's say, is. So what I can do here is highlight this region of text, right click on the actual feature itself, and click Remove Sequence from Feature. This is going to appear as a gap, both on the sequence and on the actual map itself. And importantly, if I decide to copy this feature and paste it into a different project, the feature sequence, the feature title, the gap, any notes, they can all be copied over as well to make sure there is a high level of consistency. So we can see how all of these features and how the sequence looks kind of in front of us. But one important thing is being able to see how this is going to look in our target cells. So we can do this by turning on our translations here. Now, VectorBee is going to make an approximation, primarily based off of relationships with the number of nucleotides and start codons. But if we turn this on here, typically VectorBee does a really good job of just automatically assuming where this is going to be and being correct. I have found one instance here where we need to make a slight adjustment. And luckily, that's incredibly easy. So we go to this EGFP right here. We can just right click, click Edit Feature, and then we can ensure that this feature is translated in frame. So we can either do a +1, +2, or +3 frame. Just make sure that's on, click Save, And we can see that this new little line has appeared for our coding sequence. Now we can change how this looks either in terms of the amino acid name or the color. Here, to just really highlight this coding sequence for myself, I'm going to color those amino acids. So now that we have those colored amino acids, fantastic, all set. So we have our vectors organized exactly how we want them. We have everything ready to go, but we already have a number of vectors that are open. We're starting to make some changes. We're going to be making some more edits, and it can be hard to keep track. One of the first things, of course, we can do is just make sure we're saving our progress. So I'll go ahead and save this file here. It's just a vectors today file. But we have far greater organization capabilities when we go to our project dashboard here. This is your hub of organization. And first thing, you can view all of your vectors on this sort of global scale. But additionally, you can do things like sort and search. However, my favorite thing is this little Create Project Binder button here, because this allows us to do a lot more organization. I'm going to make a folder here to actually create a hub for my CRISPR vectors. I'm going to change the color so that it matches my fingernails today. And then I'm going to select which vectors I want to go into this binder. Now I have everything all set up in this little binder, in this folder. I can move things around. I can change visibility, but I can also change individual project information. Here, I like having my vector ID over here, but I wish I had a different name. So what I can do is right click. I have a number of editing options. Here, I'm just going to click Edit Project Information. And because I want to keep my vector ID in my vector map, I'm going to paste that down here and just call this something more recognizable. So SOX2 gRNA1. And now we have our vector ID still, but we have a new name for that project. And once everything is organized exactly how we want, we can leave the project dashboard by clicking this X right here. And we can see that we have this new name for the project, but our vector ID is still present. So everything is all set up and ready to go. So our visualization is done, our organization is done, but that's really just a pretty small part of what we want to do in VectorBee. What we want to do is create our ideal vectors and get them in our hands pretty easily. So in order to do that, we need to start editing. Now, this is very straightforward in VectorBee. One thing that we're going to start with is something pretty straightforward. And actually, I mentioned it before. So what I want to do is go back to that control vector, and I want to replace the EGFP in that control with SOX2. One easy way to do that is just click on this feature and press delete. That's going to remove that EGFP. It's going to ask us if we're sure, yes. And then we can go to where we want to insert that component. We're going to right click. We can either insert sequence or insert component. But I want to note another way that you can do this. By clicking Command Z or undo, we get our EGFP back. And when we click on that feature and right click on the sequence, we can replace with sequence or replace with component. Now here we see the VectorBuilder database of components that is huge, it's massive. Right now, I'm really just gonna focus instead on this refseq ORF because I just want a SOX2 transcript. So I'm gonna search for SOX2. I have a number of different species that are returned, but I want the human refseq ORF, great. And I can double check my sequence here if I have that information handy, but it's a lot easier to just double check this refseq protein ID or refseq transcript ID. Now I can add this, and then I have extra kind of options for customization of how this feature appears. I'm happy with all of this, so I'm just going to click Insert. All right, and then I have my SOX2 that is just downstream of that COSAC in place of my EGFP. I've turned my control vector into a SOX2 overexpression vector. But let's say I'm not done. Let's say I want to also add a tag onto this. First thing I'm going to do is go ahead and quickly make sure that this feature is being translated in frame. So I'll just do that quick little step to make sure. And then what I can do to add a tag onto the end of this sequence is just go to my vector map, click the relevant region, and then go to the end of my sequence, click where I want to add that tag. Again, just simply right click, click Insert Component, and then go over to the region with the tags. We have a vast number of tags that are organized already into different components or different types. And then I'm looking for just a purification tag. So I'm going to add this 6x his tag onto the end of my protein. Go, and yes, we can double check that sequence and click Insert. All right, and now I have my tag at the end of my SOX2 sequence. Fantastic. However, because I have translations on, I can see that I have this stop codon. So I can easily delete that stop codon just by clicking that delete button. Am I sure? Yes. And now I can double check that my translation is going to go all the way through, or my translation is going to have that tag. Now, one thing is that it goes a little too far, so we want to be sure that we add that stop codon. Now, as soon as you start typing, this insert sequence is going to come up, and you can just insert that sequence, insert your stop codon, and you are ready to go. You have exactly the sequence that you were hoping for in that entire component. Now, this is a very easy and straightforward way to insert one of the many components that we have in the VectorBuilder database. However, I do want to show one other way that you can add a little bit more context or have a little bit more flexibility. So let's say we want more information about that SOX2 before we add it. One thing that we can do is go to File, go to Import NCBI gene, and then we can search for our gene. So let's search for SOX2 here, and then we can bring up those results. Let's look at what's under human, and we have one transcript. If we view the transcript table, what we see is something similar to what we saw before. We have the option for both a coding sequence and a transcript. But what I really want to focus on here is the other action, add refseq gene from our genome. So if we click this, it opens up as essentially a new project in our entire VectorBee window. And what we have here is all of the information that NCBI hands over, but in a more organized way. So we can see the entire gene, we can see transcripts, we can see regulatory regions, we can see the coding sequence, promoter, enhancer, and other features that NCBI has imported into all of this information. So let's say that I look at all of this and I say, yes, this is what I want. I have this within my relevant vectors. I have the sequence that I need for these various different regions. Fantastic. I'm done with this now. I can click this little hide project, essentially an X, but it's not really an X, because if we go to our project dashboard, our X'd out file, essentially project, is still there. If we're completely done, we can delete it, but we can always bring it back. It is just kind of a nice backup, a nice way to ensure that just because you X out of something means you don't want it right then, but you might want it later. And we want to be sure that you have the option to bring that back should you need it. So we have our vectors all sorted out. I have my overexpression vector here. I have my other CRISPR vectors. They're organized, fantastic. I'm ready to go, right? In the past, the next step would be try to figure out the cloning method. See if you can use restriction enzymes. Start looking into other cloning approaches. Best case scenario, you start looking at your various different restriction enzymes that you have in the lab. You also start to download hours and hours of podcasts because you need something to keep you company while you sort through all of these different vectors and try to figure out how you're going to combine them or run PCRs to get the relevant sequence. All of these sometimes simple, but sometimes not, often very finicky cloning approaches. Now, luckily for VectorBee, you don't have to worry about that. Once you've made your ideal vector, or vectors, all you have to do is go to File and click Send Service Request to VectorBuilder. This is going to bring up this window where you can select either entire binders or individual vector projects to send over to the VectorBee team. When you click Submit to VectorBuilder, this will bring up a separate window that already has that file loaded up. So you can just input any other comments or questions or considerations that you want to keep in mind, and you can have everything ready to go. The service team can get back to you, and you can have those vectors in your hand, as well as any downstream services like packaging or library preparation. Now, we do want to be sure that you have the option to do those downstream analyses, of course. So if you do want to perform, let's say, a restriction digest, then we can easily do that by just clicking on these enzymes up here and clicking Select Enzymes. We have a large list of enzymes that you can choose from, so you can either search popular ones or kind of scroll through this list. Here, the red enzymes are ones that are non-cutters, the bolded are single cutters, and then all of the others have the number of cuts in parentheses after the enzyme. So you can also filter by the sequence recognition type, by the breaking point type, but we also have a number of groups here that you can choose from in combination. So here I want to see all of the single cutters as well as all of the double cutters. So I can click OK, and this will bring up the list of all of the enzymes that cut once or twice. Now from here, I can just select the enzymes that I already have in the lab. So I can choose these, XBA1, and then I can save this as an enzyme group just for future use. So I'm going to save this as CRISPR cutters. And then I can apply this to this project right here. Now I can see where each of these enzymes is going to cut, both on the map and on the actual sequence itself. And importantly, I can hide or show those cutters if things are starting to get a little messy looking. And next to each of these enzymes, I can actually see the sort of pattern that it will cut in, those little icons next to the enzyme. But if I want to actually perform a restriction digest, I just go right back up to this enzymes drop down, click Simulate Enzyme Digestion, and then I can either search fresh or I can select from all of those enzymes that I just saved. So I can scroll all the way down to CRISPR cutters, and I want XBA, SFI1, and SPE1. So from here, I can just submit this, and I will get a basically readout of the different fragment sizes, as well as, if you click View, a view of that vector map with where you are going to get those different cuts and the fragment sizes that this is going to produce. There we go. So we have our restriction digests that are easy and straightforward. We can also create custom enzyme groups based off of the enzymes that you have currently available. Now, in addition to producing restriction digests, another very common analysis that you're going to want to do is design primers. So we have a couple of ways to create or design primers. Over here, the difference between these two is basically how much control you want. So if we want to create primers for this gRNA, we're very restricted here because of the size. So let's say that I want to create primers with this specific sequence right here. Now for this sequence, I have 20 nucleotides right here, which I can see over in my little heading up here, 20 nucleotides. I can check my GC content. I can also check the melting temperature. And if I'm happy with all of those conditions, I can just go back up to primers. I can click Create Primer, and then I can name this just simply Primer 1. I can specify whether it's top or bottom strand. I can change color. I can also note all of these attributes that we mentioned and do things like add notes. So we have our first primer. I can create a primer on the bottom strand as well, which is very useful if you, again, really want that tight control. But if there are times when you don't necessarily want to decide on the primers themselves. For instance, if you just want confirmation, if you just want to perform a PCR where you're confirming that an insert is there, or if you're designing a probe for in situ hybridization, then you don't really care where the primers are necessarily. So you just want them to be in the right location. So if we have this Cas9 Neo complex, if I want primers just to show that this is here in my vector that I'm working with, I can click Design Primers. Now my template sequence can be what I've selected, or it can be the entire vector. And from here, I can make a number of adjustments to settings. I can change the included region. This is relevant if you kind of chose your entire vector as the selected region. You can also change the target region, so that's stuff that has to be included. So the primers are always going to be outside of this region. You can also choose excluded regions, and you can change primer features like the size. You can change the melting temperature, the GC content, and change the product size. Now for this particular instance, I'm happy with all of these default options. So I'm just going to go ahead and say the primers can be designed for me. Let's go for it. So I can click Design Primers, and primer pairs will be read out in forward and reverse. So they'll be kind of paired together. And it shows our location, kind of where we are in our sequence, the sequence of the primers, GC content, melting temperature, length, and product size. For this case, I like those first two, so I'll choose these two. They're kind of toward the end of my region of interest, so I'll add those primers here. And we can see our primers on both the vector map as well as on our sequence map. So we can kind of check where they are, double check that we're happy with the product that that's going to produce. And we can also edit this primer if we want it to be named something a little nicer than that. So new primer 1, we can save that. There we go. And then we can ensure that all of this is showing exactly what we intend. So we have our analyses for our restriction digest. We can design our primers. One thing that I wanted to mention is that we have multiple different tools, including different alignment tools. And for now, I'm really going to highlight our multiple sequence alignment. We also have various different pairwise options, but our multiple sequence alignment is one of our newest offerings and is only available through VectorBee currently. So if we select our multiple sequence alignment, we have a few different ways that we can input our sequence. So I'm going to use this method 2 because this is using vectors and sequences that we already have open in VectorBee. So first of all, I'm going to select the 3 CRISPR vectors that I had in that little binder for my SOX2 CRISPR experiments. I could enter a DNA sequence, but I just produced this nice little SOX2 overexpression plasmid. So I'm going to go ahead and import that. Now you can state a reference sequence, but I won't worry about for this. I'll let the tool kind of decide its own consensus sequence. And what we get as a readout is usually a pretty zoomed in view. And if we zoom out, what we can see are all of these regions in gray where there is kind of consensus. The red regions are the mismatches. And what we can see are these top 3 are very, very similar, except for this one little region in red, which is not surprising. These are all CRISPR vectors that all contain the same backbone, the same Cas9 Neo. It's this gRNA sequence that's the major difference. But it's kind of hard to tell that that's where the difference lies because you're just looking at this raw sequence, especially compared to that 4th vector. So what we can do is we can, instead of just seeing the raw sequence, we have a few different ways that we can change visualization. But here we're going to view the features on all of our different sequences. So now we can see that we have good consensus among those promoters. Our gRNAs are exactly where those mismatches are occurring as you would expect. We have a high amount of overlap with this neomycin resistance gene, and we can see these other regions of our vector that are very, very similar, considering they are especially all lentivirus vectors, but they're all going to have some of those same basic components. So our sequence alignment is all set up, and we can actually view this as an individual project. So this, again, similar to our NCBI search, is totally a separate project that we can go into our project dashboard to view separately. We can hide it, we can delete it, we can edit it as much as we want to, and we can come back to it as much as we need. So all of these visualization, design, analysis, editing tools are things that we have been working incredibly hard on with VectorBee to make it a go-to DNA software for everything from start to finish, from design all the way through production to analysis. So you can visualize a number of different things for your map and your sequence, and you can customize that visualization. You can annotate and edit your features. You have a number of different maps that provide those useful resources. You can make unlimited edits to unlimited regions of your sequence. You can organize and design a number of different analyses. And at the end of the day, you can utilize VectorBuilder from start to finish, from picking your vectors all the way through to actually creating your ideal vector that comes to you without needing to spend time at the bench performing those experiments yourself to just get this reagent. Your reagent can come to you and it can be exactly what you designed. So thank you all very much for your time. I greatly appreciate it. And I would love to hear any questions.